we know that all living things need food, water, and air to survive. The oxygen provided by respiration is used to convert food into energy. While humans use their respiratory organs to take in oxygen, plants have no such specialized organs. This is because plants utilize less oxygen for the oxidation of food. Plants utilize only a small amount of food for oxidation, and the rest is used in building their body, in the form of cellulose. Further, the demand for oxygen is supplied during photosynthesis, where oxygen is an end product. Plants respire through small openings called the stomata on leaves, and lenticels on stems and roots. Unlike animals, each part of the plant takes care of its oxygen needs. Plants respire at a slower rate as compared to animals. In the woody stems, the living cells are present close to the surface and aid in the diffusion of gases. Diffusion of gases is also facilitated by the presence of loosely arranged parenchyma cells that provide air spaces. Next to respiration, the requirement of food becomes an important thing for survival. Humans and animals are heterotrophs, in the sense that they obtain food from plants and other animals. Saprophytes like bacteria and some fungi feed on dead organisms. Green plants and cyanobacteria are the only organisms that can synthesize their own food using photosynthesis. They are the food source of the world, as animals, either directly or indirectly, depend on them for food. The superficial layers of plants contain cells that possess chloroplast, which aids in trapping sunlight to manufacture food. In this process, light energy is converted into chemical energy and stored in the bonds of carbohydrates like glucose, sucrose, and starch. All other non-green parts such as woody stem and roots don't have chloroplast, and hence they have to be supplied with food from leaves. The process of breathing is connected to the oxidation of food inside the cells. While breathing involves gas exchange between organisms and the environment, respiration involves the breakdown of the C-C bonds of complex compounds of food using oxygen to release energy inside the cells. In eukaryotes, it takes place in three distinct stages, namely glycolysis in cytoplasm, Krebs cycle, and the electron transport chain or ETC in the mitochondria. Since it takes place inside the cells, it is called cellular respiration. The oxidized compounds are called respiratory substrates. Carbohydrates are the usual substrates for cellular respiration, but proteins, fats, and other organic acids are also used by plants in certain conditions. During the oxidation of respiratory substrates, energy is not released directly in the cell. Instead, it is released slowly in a series of steps controlled by several enzymes. While a small part of the energy is released as heat, most of it is used for synthesizing adenosine triphosphate or ATP, a high-energy molecule. ATP is utilized for carrying out all the activities such as absorption, transport, movement, reproduction, and breathing. Compounds like NADH formed during the oxidation of respiratory substrates are utilized in the ETC to derive energy in the form of ATP. The carbon skeleton obtained during respiration is used in the synthesis of other biomolecules like nucleic acids. When the oxidation of food substances takes place in the presence of oxygen, it is called aerobic respiration. It yields water and carbon dioxide as the byproducts. This type of respiration takes place in humans, plants, and some bacteria. When the oxidation of food substances takes place in the absence of oxygen, it is called anaerobic respiration. It yields alcohol and carbon dioxide as the byproducts. This type of respiration takes place in anaerobes and archaea. Anaerobes are divided into facultative anaerobes and obligatory anaerobes. Examples of facultative anaerobes include Escherichia coli that can survive in the presence as well as absence of oxygen. Examples of obligatory anaerobes include Clostridium tetani that survive only in the absence of oxygen. All living organisms contain cells that derive energy by breaking down nutrients like glucose during the process of respiration. The energy thus released is trapped in the form of adenosine triphosphate or ATP. The energy currency of the cell. The breakdown of nutrients takes place with the help of enzymes that catalyze biochemical reactions. Glycolysis is a 10-step process that takes place in the cytoplasm of cells. It takes place in both aerobic as well as anaerobic organisms as it doesn't involve oxygen. It was first identified in yeast cells and mammalian tissues by Emden, Meyerhoff, and Paranas, and hence is popularly called the EMP pathway. The term glycolysis is derived from the Greek words glykis, meaning sweet, and lysis, meaning splitting. It involves the cellular degradation of glucose to pyruvate or pyruvic acid. In plants, sucrose, obtained from photosynthesis, is converted into glucose and fructose by the enzyme invertase. The glucose thus produced is utilized in the glycolysis process. Glycolysis takes place in 10 steps involving 2 phases, preparatory and payoff phase. The preparatory phase starts with the phosphorylation of 6-carbon glucose using ATP to form glucose 6-phosphate. It is catalyzed by the enzyme hexokinase. Glucose 6-phosphate thus formed isomerizes to form fructose 6-phosphate. Fructose 6-phosphate is then phosphorylated using ATP to form fructose 1,6-biphosphate. The fructose 1,6-biphosphate splits into 2 molecules of 3-carbon 3-phosphoglyceraldehyde or PGAL and dihydroxyacetone phosphate or DHAP. As DHAP can't be utilized directly in the payoff phase, it isomerizes into PGAL. Thus, 2 molecules of PGAL are formed from 1 molecule of fructose 1,6-biphosphate. These reactions complete the preparatory phase of glycolysis. The payoff phase starts with the phosphorylation of PgAl using inorganic phosphate to form 1,3-biphosphoglycerate or DPGA. The two redox equivalents removed from PgAl are transferred to NAD+ to form NADH2. The DPGA thus formed undergoes dephosphorylation to form 3-phosphoglycerate or PGA. This is an energy-yielding step as the phosphate released from DPGA is captured by ADP to form ATP molecules. As the inorganic phosphate is directly transferred from a substrate to the ADP molecule to form ATP, it is called substrate-level phosphorylation. The phosphate group in 3-phosphoglycerate is relocated to form 2-phosphoglycerate, which is then converted into 2-phosphoenol pyruvate or PEP by losing a water molecule. The final step in the payoff phase is the energy-yielding reaction involving dephosphorylation of PEP to form 3-carbon pyruvate. The inorganic phosphate released from the PEP molecule is captured by ADP to form ATP molecule, leading to substrate-level phosphorylation. This ends the process of glycolysis. Of the 10 reactions, the 3 involving glucose, fructose 6-phosphate, and phosphoenol pyruvate as substrates are irreversible, while the others are reversible. As 2 molecules of PgAl are formed from fructose 1,6-biphosphate, the reactions in the payoff phase will occur 2 times, once for each PgAl.